Very good afternoon to all of you. So today we will have a seminar from one of our research scholars, Mr. Jadesha. He will be giving a seminar on the topic, the epidemiology and management of Phytophthora Blight of Pigeonpea. This Phytophthora Blight is an emerging disease in Pigeonpea, and more Mr. Jadesha will talk about his work, what he has done in his tenure of this Ph.D. And I invite Jadesha to come here, and Jadesha, you are requested to complete your seminar in 40 minutes, and then we will have a discussion for another 15 to 20 minutes. So Jadesha.
Thank you, ma'am. Good afternoon to all, and it's my immense pleasure to welcome you to my thesis seminar on epidemiology and management of Phytophthora Blight of Pigeonpea. Pigeonpea is an important component of cropping system in the semi-arid tropic, and the Pigeonpea is the second most important food legume after chickpea. Among the biotic stresses, Phytophthora Blight is recently gaining importance, and this is the third most important disease after Fusarium wilt and the Stellate disease. Though disease they reported in 1966 from New Delhi, emergence of occurrence of disease is in recent years, and it is causing the huge grain yield losses. And information related to biology, epidemiology, and management of Phytophthora Blight disease is limited. In this background, following objectives are framed for my thesis, Ph.D. thesis research, and objectives are assessment of spatial and temporal incidence of Phytophthora Blight of Pigeonpea, morphological and physiological categorization of the Phytophthora drechsleri foreign species Cajani, the study of epidemiological factors of Phytophthora Blight of Pigeonpea, and lastly management of the disease. To assess the spatial and temporal incidence of the disease, pixel plot survey and roving survey was conducted in ICRISAT and Deccan Plateau of India. To assess the occurrence of the disease across the location, we have conducted a roving survey, and to assess the incidence of the disease with respect to time in a season of a particular locality, we have conducted periodical survey. Periodical survey was conducted during Kharif season in 2012 and 2013, and the survey was conducted at a regular interval at 30 days, 30, 60, 90, 120, 150 days after sowing. And in 2012, we have made survey of 16 Pigeonpea fields at ICRISAT location. Among the 16 Pigeonpea fields, 3 fields they have showed the incidence of Phytophthora Blight, and the incidence was ranged from 15% to 59%. In BP15B has showed the highest incidence. And during 2013, we have made survey for 21 Pigeonpea fields, and among all the fields, BP14C has showed the highest incidence of 55%. And periodical survey. Periodical survey showed that the incidence was occurred throughout the cropping system, initial from seedling stage to maturity of the crop. Main objective of the periodical survey is to assess the incidence of the disease with prevailing weather parameter. These are snapshots showing incidence of Phytophthora Blight in BP14A field, and this is the ICPL 20092, and Kamika they have showed incidence of up to 100%. This is a snapshot showing during early stage of during 90 days of the 90 days after sowing. And these two snapshots showing at the end of the season. We can observe here at the mid-season almost 60 to 70% of the plants are infected. And at the end of the season, all plants are uprooted out, and these all are infected with Phytophthora Blight. In these two varieties, the incidence was 100%, and they have taken sowing of 3 rows, and each row containing about 60 to 70 plants. All plants are infected with Phytophthora Blight. And this is a wild relative of Pigeonpea, Cajanus scarabaeus. This is also showed incidence of more than 80%. All plants they have infected with PB, they have died off. And one more snapshot showing incidence of the PB at at least at 90 days to 100 days old plant. After the at the end of season, all plant they uprooted out. And this is one more photo they have taken. We have taken at the other BP14C, and where all plant they uprooted, and all in this field 55% of incidence was observed in 2013. And the major objective of the periodical survey is we are correlating the incidence of the disease with prevailing weather parameter, and we have correlated the weather parameter, cumulative rainfall, average temperature maximum and minimum, and average relative humidity of maximum minimum. Among all weather parameter, rainfall and relative humidity maximum has showed the significant positive correlation, whereas the temperature minimum and relative humidity minimum has showed the negative correlation. In this case, disease will incidence will be more as rainfall and relative humidity is increasing. Where on the other hand, if incidence will be less as temperature and relative humidity, reducing temperature and relative humidity. To determine the extent of correlation, we have calculated the multiple regression analysis, and equations show that here y is then a dependent variable, our percent disease incidence, and x1, x2, x3 are the independent variable. How much these independent variable contributing to the disease we have calculated out, and for the we have made for 2012 and 13 year data, it showed that y is equal to intercept of -67.20, and slope for x1 is cumulative rainfall, x2 is the average minimum, average maximum temperature, x3 is the average relative humidity, and by this equation we can predict the incidence of the disease, incidence of the disease, and by periodical survey it is concluded that the incidence of disease or outbreak of disease will be more, we can expect more during the erratic rainfall of 100 to 200 mm within a span of a week, with temperature of 27 to 33 degrees Celsius and relative humidity of more than 75%. These are the typical symptoms of Phytophthora blight in the field condition. Initially, symptoms will be observed on the leaf, and the initial symptoms will be water soaked. You can observe here, water soaked area will be water soaked area. Within a week, with a favorable weather parameter, this water soaked lesion will become to will turn into blight symptom, and a similar blight symptoms was observed on the stem portion. We can observe here the stem lesions, and initially these stem lesions will be of 2 to 3 centimeter. At the end of season, these symptoms will make increase up to 15 to 20 centimeter. And another symptom of typical symptom of Phytophthora is plant will break down at the lesion of the stem lesion, and the whole plant will dry off. And another symptom, another typical symptom is formation of the stem gall in the adult stage, and this is due to increasing in the production of oxygen. These are the typical symptoms of Phytophthora blight in the field condition. Besides periodical survey, we have made a survey, roving survey to study the distribution of the disease across the location. Survey was conducted in Kharif 2012 and 13, and in Andhra Pradesh, Adilabad, Medak, Rangareddy, and Mehboob Nagar districts. Now these districts in the Telangana, and in Karnataka, Bidar, Gulbarga, Raichur, and Ballari. In Maharashtra, we have conducted about 10 districts of Maharashtra and 1 district of Madhya Pradesh. During Kharif 2012, we have made survey of 122 farmers field, and of these 7 districts, and results are highest incidence, disease incidence is ranging from 1% to the 32.85%. Among all the districts, Akola district has showed the highest incidence, followed by Bidar district of Karnataka with incidence of 30.21%. Similarly, again in the 2013 Kharif, we have conducted survey in the 4 states, and and we visited around 146 villages in the survey of the 15 districts, and incidence is highest in the Shiva district with incidence of 15.96%, followed by Bellary with incidence of 58%. These varied incidence of the disease is maybe due to uneven distribution of the soil inoculum and weather parameters of the particular location. This is a snapshot we have taken during our 2012 Kharif survey, and this is at Akola. The here whole field almost it is a field of 1 hectare. 60 to 70% of plants are infected with TB, and this is a snapshot we have taken in the demo field of ICRISAT, and this is the clear difference between the healthy and infected and healthy plant, where we can observe the stem lesions on the plant, and this is a symptoms of stem blight symptom, and all these are plants infected with TB, and they are showing the typical TB symptom. After this stem lesions, the whole plant will dry off and it will get breakdown. From this survey, we have collected the infected sample, and we have made isolation on the V8 PARP media. Since the isolation of the Phytophthora is much difficult on the normal media, we have amended the V8 media with antibiotics, phenaricin, ampicillin, rifampicillin, and TCMB, pentachlornitrobenzene. These antibiotics will suppress the growth of the bacteria, since Phytophthora isolation and Phytophthora culture are usually more prone to contaminated with the bacteria, and we could able to success. We have isolated the pathogen and V8 PRP media, and same pathogen was studied for morphological and physiological characteristics. Culture Phytophthora will take around 6 to 7 days to complete the whole full petal plate, and culture will be like a dull white color with rosy type mycelium. Here we can observe this is a typical characteristic of Phytophthora species, and here hyphae is the synesthetic hyphae, means hyphae is without cross holes. Then here we can see the formation of hyphal swelling. This is a typical character of Phytophthora species, where we can differentiate the from pythium. And here sporangia is on terminal sporangia is formed on the sporangia 4. This is sporangia bearing structure. This is sporangia Phytophthora drechsleri Cajani. Sporangia is non-papillate and non-pedicellate sporangia. And one more typical characteristics of Phytophthora drechsleri is formation of sporangial proliferation, where sporangia is formed from the previous side of the sporangia. Here we can observe the formation of new sporangia from the site of old sporangia, and this is a typical characteristic of Phytophthora drechsleri. And another characteristic of drechsleri is sporangia is intact to the mycelium, where it won't shed after the sporangia, after liberation of juice spore. Here you can see the empty sporangia, and sporangia will form on the simple sympodial branching type, where sporangia will form one alternative to other. And after the juice spore, after the liberation from the sporangia, juice spore is encysted juice spore, means it will shed its flagella and it will get encysted within 15 to 20 minutes. And this Phytophthora drechsleri Cajani is an homothallic, since both male and female gametangia are formed on the same mycelium. And here you can see this is oogonium, this is antheridium, and here sexual reproduction is amphigenous. Both male and female gametangia are perpendicular to each other, and this is a sexospore, oospore. And the pathogen was studied for physiological characteristics. Here we have conducted experiment to determine the influence of media and temperature and relative humidity and carbon dioxide on the growth of the pathogen. We have used the 12 different media to determine the to determine the efficacy of the medium to support the growth. And among the 12 media, tomato juice agar media has showed the highest growth, followed by V8 juice agar, then potato destroys agar. And these three agar media we have checked for mass multiplication of pathogen and the broth condition, and the results showed that both V8 juice broth and tomato broth has showed unfair result. And the tomato juice agar, it will require 7 days to complete the whole petrolate. And hence this is the photograph. We can see the growth of the pathogen on different media. This is a maximum growth on the tomato juice agar on the 7th day after incubation. The effect of temperature, and temperature was studied at different incubator condition. Here from different 8 ranges of temperature was maintained in different incubator condition: 5, 10, 15, 20, 25, 30, 35, 40 degree centigrade. And here we can observe the growth of the pathogen was nil at 10 degrees Celsius, at 40 degrees Celsius also. And the rate of growth is increased up to 30 degrees Celsius. Later it has decreased. And among all the temperature range, 30 degrees centigrade is the optimum temperature to induce the growth of the pathogen under in vitro condition. And similar experiment was conducted to determine the influence of relative humidity under incubator condition. Here growth of the pathogen was observed on the all relative humidity ranges, and but increased growth was observed at more than 75% of relative humidity, and it is showed, and similar experiment was conducted on the liquid broth. In liquid broth, it showed that 100% relative humidity has supported the maximum growth of the pathogen. And from the relative humidity, it is concluded that growth of the pathogen is high at 100% relative humidity. Similarly, as now climate change is becoming a major threat for agricultural production, we have conducted an experiment to determine the influence of elevated CO2 condition on the growth of the pathogen under in vitro condition. Here we have maintained the three different CO2 concentration at incubators. One is at ambient CO2 concentration 350 ppm, and the two elevated CO2 concentration 550 and 700 ppm. And results show that means at elevated CO2 condition, it is aggravating the growth of the pathogen. And where we have observed the 90 mm growth at 6 days after incubation itself, where it is ambient showed the 80 mm. And the growth of the pathogen also varies with the varied CO2 level, where we can see the smooth margins at the ambient CO2 level, with whereas in irregular margins at the elevated CO2 levels. And this is a positive. This is inferring that at elevated CO2 condition, pathogen growth of the pathogen is rigorous, and where we can expect to increase disease incidence at higher CO2 levels in the field conditions. From the physiology of the physiological studies of the pathogen, and we have not observed any sporangia and juice spores at different media temperature and relative humidity level. Hence, we have made attempt to develop the separate protocol to induce the sporangia and juice spore under in vitro condition. Here we have tried with the different protocol where they have reported against the different species of Phytophthora, and none of the protocol has induced the sporulation. Hence, we have developed the protocol to induce the abundant number of sporangia and the juice spore. This is a related tomato broth method. Pigeonpea seed for medium also showed a sparse amount of sporangia and anthelidium, ugonium, and oospore. In this protocol, we have used the diluted tomato juice broth where we have taken 50 ml of tomato extract and we made to the volume of 1 liter diluted broth. Here we have to select the 6 days old culture, and from the margin of the pathogen we have young mycelium we have transferred to the Petri plate, which is containing diluted tomato juice broth, and we have incubated at 25 degrees Celsius for 24 hours. It alternates 12 hours of light and dark condition. After the 24 hours of incubation, we can observe here growth of the mycelium. This is young mycelium. After the 24 hours, we have replaced the broth with the sterile distilled water, and we have again incubated for 24 hours. Later, after 24 hours, we can observe the formation of sporangia and juice pore at the margin of the mycelium. This is an sporangia, and this sporangia is liberating the juice pores. And we have also observed that after 48 hours of incubation, there is formation of antheridium and oogonium and oospore also. Since Phytophthora drechsleri Cajani is not producing sporangia and juice pore in under protected condition, this is a protocol we need to use to study the morphological and physiological characteristics of the pathogen, and it is also helpful for the inoculation of the plants under in vivo condition, and to study the interaction between the pathogen and this protocol can be used to induce the juice pore and sporangia. This is the different snapshots we have taken from that protocol, and here we can see the mature sporangia, where sporangia is liberated the juice pores. These juice pores are embedded in the membranous sac of the sporangia. When the membranous sac is burst, these are frames. They are showing the liberation of juice pores in sequence. Here you can observe as the juice pore is emitting out, and finally there is an empty sporangia. And we have, since facilities are available with good microscope and camera, we have taken video of release of juice pore. This is a video. Here we can see the release of juice pore from the sporangia. Around 100 to 120 juice pores are released from each sporangium. These juice pores are biflagellate and motile. These are having a tinsel and a tail type of flagellum. And after juice pore release, it will take 15 to 20 minutes to shed this flagella, and it will get encyst. And after we have taken and we have observed the growth of the juice pore under in water condition. Here we have taken the juice pore on the slide, and we have observed for the growth of the juice pore. This is we can see the growth of the juice pore. And finally, since absence of host, the juice pore has desiccated. You can hear fragment of the mycelium. You can see here, and it has taken around 1.5 hour time. Since if a host will be there, then it will form the aprosorium to infect the seedlings plant. And similar, this same protocol we used to determine the influence of weather parameter and the induction of juice pore. Similar experiment protocol we have used under the same protocol we have used, and here we have changed the different temperature levels at incubator condition, and temperature showed that we have used the 20 to 40 degrees centigrade, and among all the temperature range 30 degrees centigrade has showed the highest number of juice pore induction, and the similar experiment was conducted in the relative humidity. Here number of juice pores are increased, they are increasing relative humidity, and you can and CO2 condition, elevated CO2 condition is having positive impact on the induction of juice pores. Then we have made experiment to determine the influence of weather parameter on the host pathogen interaction. Here you can see the epidemiology is very much important. Near host pathogen interaction without conducive environment, there is no disease. If with conducive environment, you can see the disease. And here weather parameter and adaptive factors we have taken, we have considered to check the influence of the host pathogen interaction. We have temperature, radiative humidity, and carbon dioxide, and the soil type and soil moisture. These weather factors we have conducted experiment in the incubator condition, whereas soil type and soil moisture experiment we have conducted in the glasshouse condition. In all the experiment here pathogen we have used the susceptible variety ICP 7119, and pathogen was artificially inoculated. And here under among all the temperature ranges, there is no incidence of the disease at 5 and 10 degrees Celsius, where we can see the increased incidence of the disease from the 15 degrees Celsius, and highest incidence was observed from 25 to 40 degrees centigrade. Here we can, whereas incubation period is inversely proportional to the incidence of the disease. With here incubation period is time lapse between the inoculation and observation of the initial symptom. As the temperature is increasing, the incubation period is decreasing, and here you can observe the incidence of the disease even at 40 degrees Celsius. Since the aerial temperature is 40 degrees Celsius, we have checked the your soil temperature is very less. Since we are watering the pots 3 to 4 times in a day, temperature of the soil is around 32 to 33 degrees centigrade. Hence, this may be the reason that we have observed the incidence of the disease even at 40 degrees Celsius. And it is a similar experiment was conducted to determine the influence of relative humidity. Here we can observe the incidence of disease from in all the range of relative humidity, but increased incidence of disease at increased relative humidity. Since this may be region that pathogen will sporulate high in the higher relative humidity. And we have made another experiment to determine the influence of CO2 condition in the under OTC, open top chamber facility. Here OTC 1 we have maintained the ambient CO2 condition 350 ppm. At OTC 2, OTC 2 and 3 we have maintained the elevated CO2 concentration 550 and 700 ppm. We have inoculated the plants with both in soil mixing and soil drenching method, and the incidence of the disease is high at elevated CO2 condition in both the both the inoculation method, and we have observed the 100% incidence on 7th day after incubation. This is the photograph showing the incidence severity of the disease. This is control plants. And here plants they show infected plant at 350 ppm, this is 550 ppm and 700 ppm. You can here observe the severity of the disease more in the elevated CO2 condition than ambient CO2 condition. Since elevated CO2 condition is positively influencing the growth and sporulation of the pathogen, this may be reason that we can see the increased severity of the disease at elevated CO2 conditions. After weather parameter, we have also conducted experiment to determine the influence of adaptive factor on the incidence of the disease. And here experiment was conducted to determine the influence of soil type. We have maintained the two black soil and red soil with the two different inoculation methods. Here incidence was highest at the red soil with soil mixing type of inoculation method, and followed by red soil with soil drenching method, where we can see the maximum difference of between the red soil and black soil. Highest incidence of 93.3% incidence we have observed on 10th day after incubation, whereas highest incidence of 43% we observed in black soil condition. Hence, it shows that red soil will favor the incidence of the disease. And experiment was conducted to determine the influence of the soil moisture, and here a different soil moisture level was maintained in the pot under pot condition. Here we have maintained the 60%, 80%, 100% field capacity and flooding condition. This 60%, 80% and 100% field capacity we have maintained by using the gravimetric method. Before maintaining, we have calculated the initial soil moisture and available soil moisture content of the soil. Hence, we have determined how much amount of water we need to add to maintain the respective field capacity. And we have inoculated here two different inoculation methods, and the results showed that the incidence of disease is highest at flooding condition, and whereas 50% in 100% field condition, but least in the 60% field condition with 22.5%. And in soil, this is under soil mixing method. In soil drenching method, there is a nil incidence disease was observed up to 80% of field capacity, whereas in 100% we have observed around 15% of incidence. But in flooding condition, it has induced the 100% incidence on 10th day after incubation. And it is sure that since flooding condition will favor the pathogen for sporulation, hence we can expect the incidence of the disease with high %. This is the photograph showing the influence of soil moisture. Here, first row we have maintained the 100%. First row we maintained the flooding condition. This is soil mixing method of inoculation. These are the first 3 columns are control plant. These are the soil mixing method of inoculation. This is soil drenching method. Here we can see the all plants incidence was 100% compared to the even though 100% of field capacity incidence is very less. Then management of the Phytophthora Blight. In that we have host plant resistance. This is the reliable method to manage the disease with cost effective and eco-friendly strategy to manage the disease. And development of a Semi technique is an integral part of host plant resistance. Since Semi technique are necessary to screen the genotype. And in this aspect, we have standardized the different inoculation technique and optimum inoculum concentration, and we have studied the susceptible stage of the plant and temperature, soil type, soil moisture. We can you have to maintain the optimum temperature, soil type, and soil moisture condition. We have used different 5 different inoculation techniques. Among the 5 different inoculation techniques, soil mixing method has showed the best inoculation technique to induce the disease, followed by soil drenching method. And whereas spraying of mycelium inoculum has not induced any disease, and whereas soil stem inoculation method has induced the incidence of 50.8%, and stem stacking has induced the disease of 46%. Here we have used the 2 different isolates, PD1, PDC2, and both isolates are at par in virulence. Since soil mixing and soil drenching methods are efficient, we have used these 2 artificial inoculation methods for my epidemiological studies. And from 2 effective inoculation methods, we have standardized the optimum inoculum concentration to induce the incidence, and we have study was conducted with 6 different inoculation concentrations. 1% of inoculum to the soil weight to 12.5% of inoculum to the soil weight, and the incidence was observed. The nil incidence was observed in 1% and 2.5%, whereas 5% has induced a disease of less than 10. It has showed incidence of less than 10%. But highest incidence was observed on 10 and 12.5% of the soil inoculum. Since both 10 and 10.5% of soil inoculum is showing the same incidence, and we have taken 10% of inoculum to the soil weight for all the experiment for artificial inoculation. After the inoculation technique and inoculum concentration, study was conducted to study the study conducted to determine the susceptible stage of the plant. Here we have taken 10 different stages of the plant, from starting from 10-day old to the 120-day old, and here we have taken the showing at different days, so that at final day we have obtained all different stages of the plant. Then we have made artificial inoculation with the soil mixing method, and we have taken observation up to 7 days after incubation, and but none of the all stage of the plant is showing susceptible. Since for screening, instead of using for screening it is better to use 10 to 15-day old seedling, as it will require the less time and inoculum concentration, quantum of inoculum. This is the photograph. It is showing the all stages of the plant is susceptible. This is a line. It is showing. This is the control plant. These are inoculated plant. Similarly, this is a 10-day old seedling. These are a healthy plant. You have made inoculation and with the soil mixing method. And after 7 days after incubation, we can observe none of the stages show the resistant reaction. And this is the inside picture. You can see the black stem lesion and infected part, and this is a healthy portion. Obviously, then we have conducted a screening. We have done screening for 43 Pigeonpea wilt and sterility mosaic nursery. This nursery we have nursery they have showed resistant against the wilt and sterility mosaic disease. In order to doubt the multiple disease resistant line, we have checked. We have screened the 43 lines at sure condition. It is under CPR condition. Among the 43 lines, 3 lines have showed the incidence of less than 10%. These green lines are resistant reaction. Then these blue lines is showing lines are showing less than 20% incidence. These all are moderately resistant lines. Yellow lines are showing less than more than 20% of incidence with less than 50% of incidence. These all are moderately susceptible lines, and these 5 lines are showing incidence of more than 50%. These are highly susceptible lines. And these 3 improved lines, ICPL 99044, 99055, 99099 need to be screened under glasses condition and under multi-location trials to confirm the resistance against disease. And similarly, we have taken observation of different 59 breeding lines at the BP14A field, and here we have scored the incidence of the disease for to determine the resistance. And among 59, 38 lines, 38 lines they have shown the nil incidence, and whereas 3 lines they have showed the incidence of more than 80%. And hence these lines need to be checked. We need to be screened under glasses and field condition under artificial inoculation condition. Management using fungicide. Here metalaxyl is commonly used fungicide to manage the humus fungi. Along with in our study, along with metalaxyl, we use mefenexam. This is mefenexam, and we have conducted did Pigeonpea technique to determine the efficacy of the fungicide against the pathogen, and it is showed that at 2 microliter per ml of media there is an 100% incidence of the pathogen, but whereas 5 microliter per ml of media showed the 100% incidence of juice for induction. This is a photograph. You can see the efficacy of the mefenexam fungicide, and there is a complete inhibition of the pathogen at 2 microns per ml. And similarly, trichoderma we isolated from the Pigeonpea field, and we have taken this study to determine the efficacy of the trichoderma species against the pathogen by using dual culture method. And among the 5 different isolates, isolate 3 has showed the highest inhibition of the pathogen over control with inhibition of 80%, followed by isolate 4 and 5. and least effectiveness was observed on trichoderma isolate 1. This is the photograph. Here we can see the pure culture of trichoderma isolate. This is the culture control pathogen. Here you can see the dual culture where trichoderma has overgrowth on the pathogen. And similarly, Pseudomonas species was isolated on Kingsby medium, and study was conducted to study the efficacy of this Pseudomonas isolates against the pathogen. And among the 4 isolates, isolate 1 has showed the highest incidence, highest percent reduction over control, followed by trichoderma Pseudomonas isolate 4. Photograph showing this is the pure culture of Pseudomonas isolate 1, and this is the pathogen. Here we can see the inhibition of pathogen in the dual culture technique. And then we have studied the compatibility of the trichoderma with the methanexam metalaxyl, and trichoderma was compatible up to 1000 microliter per ml of the media, and where more than 1000 microliter per ml has inhibited the growth of the trichoderma. From this result, it is showed that up to 1000 ppm, 1000 microgram per ml of fungicide is compatible with the trichoderma species. With chemical fungi, we have taken the chemical fungicide and bioagent for management of the disease under gaseous condition and field condition. Here we have used the methanexam. Methanexam as a seed treatment. Treatment 1 is showing the methanexam seed treatment for 2 gram active ingredient per kg of seed, and T2 and T3 we have reduced the methanexam concentration to half, 1 gram per kg of seed and 1 gram per kg of seed with trichoderma isolate seed treatment, trichoderma 4 gram per kg of seed, whereas Pseudomonas 10 gram per kg of the seed. And T4 is we have made seed treatment with the trichoderma isolate alone. In T5 Pseudomonas isolate alone. In T6 both you have trichoderma and Pseudomonas you have clubbed each other. And T7 metalaxyl seed treatment you have done 2 gram active ingredient per kg of seed as a positive check. And results showed that there is a inhibition of the disease was highest in the treatment with methanexam, and followed by the T2 and T3 where we have reduced the seed treatment to the half, means we have used 1 gram of fungicide plus combined with the trichoderma and Pseudomonas. And here we can see the inhibition of 90% over control. And similar experiment was conducted in the field condition at RL17 experiment. RL17 field we conducted experiment, and here we can see the incidence. Lowest incidence was observed in methanexam seed treatment, followed by methanexam seed treatment with trichoderma isolate. And it is concluded that we can methanexam seed treatment can be reduce the disease of 45% over control, and we can alternatively reduce the dose of the fungicide, and we can combine the biocontrol Pseudomonas and the trichoderma with we can use it with reduce the dose of fungicide. And from farmer point of view, it is means it is to manage the disease proper sowing up the Pigeonpea need to be sown on the well drained soil, and the proper drainage need to be taken under heavy rainfall, and growing of moderately resistant varieties Asha and Maruti. Asha is resistant to both wilt and SMD, and it is also showing a moderate resistant reaction to the Phytophthora Blight. And beside these growing of resistant lines and cultural practices, it is also recommended that seed treatment of methanexam or metalaxyl at the rate of 1 gram active ingredient plus combined with the seed treatment with the biocontrol will manage the disease effectively. These are future scope for the research work, where means confirmation and validation of resistant lines we have got at a severe location at a sickler condition. These three ICPL 99044, 55 and 99, these need to be reconfirmed under gaseous condition and multi-location trials. And another area is to study the mechanism of the resistance against the pathogen and the development of weather-based forecasting system with long-term weather data with incidence of the disease, and to study the morphological, pathological and molecular variability of different isolates of the pathogen under varied climatic conditions, and to study the production and characterization of the toxin produced by Phytophthora drechsleri Cajani. And I performed my thanks to the Department of Legume Pathology Division, Grain Legumes, ICRISAT, and the Department of Plant Pathology, Andrew, and for granting the ICR Senior Research Fellowship. And I will would like to convey my thanks to Learning System Unit, ICRISAT, Library and Information Services, and Housing and Food Services.
Thank you, Jadesha. Now you have left with very less time for question and answer. Maybe we can take few questions if you have from Jadesha.
What is the basis of selecting the three different concentrations of carbon dioxide? Correct 5.
Yes, sir. Normal carbon dioxide ppm is 365 ppm in ambient condition. To determine how much influence means how much impact of elevated CO2 concentration on the plant disease.
One question is, look at the juice spore production, look at the decisions, there is a significant difference. And coming back from 550 to 700, when there is an increase, there is no difference. And correct, what would be the next concentration? There will be a significant increase.
This needs further, because this was just a preliminary studies which was conducted. Yes, probably that is required because we have seen the trend that after 550 it has come down.
Right.
So probably it needs further studies to take one more concentration to see that what will be the effect of elevated CO2 probably. And the study,
Is the need for
it's 700. In fact,
next 200 years, CO2 levels are going to reach to that level. These are the concentrations likely to improve to the next 50 years. It is around 500. There will be a concentration, there will be. As the CO2 level elevated, and growth will be instead of planted. When the plant growth will continuously increase, the pathogen will incorporate and separate. These CO2 levels are indirectly more product directly it is going to be after that will be the reason. And I have got one typical question. You said that in Phytophthora you have observed paraganous type of reproduction. Apiganous. Okay, right.
Apiganous.
That mycelial swelling is one of the characteristics of Phytophthora Cajani. Is it chlamydospore or different?
This is different from chlamydospore.
Okay.
Very initial after 3 to 4 days after culture from the 3 to 4 day old culture we can observe the hypal swellings.
Okay. So it is not chlamydospore.
No.
Okay.
So Phytophthora does produce chlamydospores.
Yeah.
looking like chlamydospore.
No, it is hypercellular.
Why only one? Why not?
No, there are much. If you see under the microscope, you will get many hyperswelling in the one image microscope itself. Since to highlight to picture, I have taken only one swelling there. Dark color. No, it means it is under water condition I have taken. Means this the culture is on the water condition.
Maybe that larger view has given would have given a better
dark
picture of the problem.
Non-petalate. Where we can not see. Drechsleri is non-petalate. Non-pedicellate also. There is no constriction at the base of the sporangium. A clear cut of sporangial pore in the base of sporangium. Drechsleri or intestine. You get empty space when I get tip. High level. While moving, it look like blue color because of flagella.
Better clarity.
You have seen in ICRISAT field, there are two varieties, ICPL 20092 and Karmic. Both the varieties have shown 90% incidence at ICRISAT field. There are two questions because you have worked with the soil type and the varieties. Are these varieties are short duration varieties? First. Second, so generally at field level we see Phytophthora more in black soils rather than in red soils at field level. So your results are showing that red soils are better than the black soils. So whether your ICRISAT data will suggest whether these varieties are short duration or short statured plant even though they are long duration varieties. And then what was the soil type in your respective fields?
ICP 20092, it is a short stature, sir, but maturity I don't know.
Okay. Okay, then even the language variety in most of the that's why they suffer more from the Phytophthora Blight. So that is agreed. So then with regard to the soil type, the general known soil type, that is black soils are more favorable. Why in your experiment red soils are showing more disease? And what was the soil type in the ICRISAT where the 90% disease was noted?
Here we have in ICRISAT field, there is in BP14C, so that is in black soil only.
Okay.
Black soil only.
Then how do you suggest, then how do you substantiate your result with the result which you got at field level?
Sir,
You have both the results. One is a short statured plant, then you have a good disease incidence in black soils. And your part culture studies are showing simply a reverse to the actually what happens at the field level. So how do you justify?
Sir, mobility of the juice pore is more in the red soil than black soil, sir.
So what is the pore size that is required for the juice pore? The black soils also have a pore space, and the juice pores have because water is there, further saturated, then the juice pores will be able to, in most of the cases, almost all Phytophthora diseases, generally they will be having more in case of the black soil, where water tension is there, and juice pores will be able to move. But the red soils, generally quickly the water will drain out, rather than staying at the upper layers, even though they have more pore space. They will have less disease. They will have less disease. Right? Then you have to suspect it tomorrow to your exam also. Your results are, when if the results are contrast to the part of the study.
Field that both incidence at both levels, both red soil and black. You have surveyed 15 plants or 13 plants, you have to
and you got the disease only in 3 plots. And whether all those plots where you have shown the disease to us are they black sites or red sites? Because 3 or 4 plots most of them are black sites. Most of them are black. So, ICRISAT also you
So ICRISAT, no, sir.
in the red side.
In ICRISAT, we observed incidence at both the cell conditions. We have, no, sir. In that RCE23A, DHF04 and RCW01 and RL17.
Even though the disease is very less, about 50% of the number in this like that. Soil you just that's the answer.
No, in,
Because in glass house we maintain that soil very well. I think it is not possible to maintain that.
in
He has got an answer.
yes, yes, sir.
He has got an answer. So you just search out the reason for these happenings, field versus glass.
If no, then we would like to thank Jadesha for the seminar, and especially I would like to thank Professor Narayan Reddy, who is the chairman of his advisory committee and has come from the university to attend this seminar, and Dr. Pushpavati also and Dr. Kishore Verma for coming here and sparing the time to attend the seminar. Now, I request all of you to join me in giving a certificate of the completion of the thesis to Mr. Jadesha. And I request you to come here.
Thank you.
She's coming?
Come ready.
Okay.